Our group consists of myself, Luke Hvass, Chester Ashong, Catherine Bowie, Tim Schulte, and Trenton Powell, and today we'll be talking about our product, Echinacea. Viruses are small, infectious agents that are required to infect host cells and utilize host replication mechanisms in order to reproduce. Virus particles are typically helical-based protein structures that contain genetic material in their nucleocapsid, such as RNA or DNA, that they can use to infect their host cell and cause gene transfer. Sometimes viruses will also have an external envelope made up of the host cell-derived lipid bilayer, which contains proteins and sugars and gives them protection against chemicals and enzymes. One of the virus's hallmarks is that it cannot replicate without a host that it can infect. The virion will have to find a matching host cell with the correct binding proteins, as well as matching lipids and carbohydrates in order to be able to fuse with the plasma membrane and penetrate into the cell. Once inside the host cell, it can uncoat, begin replication utilizing host cell machinery, and begin synthesis of virus cell proteins. Once replication is complete, assembly of the new particles must occur. Once the key parts are assembled, the virion can leave the host cell through lysis, exocytosis, or budding and maturation, where it can then infect the host.
Because viruses cannot survive on their own, they must replicate inside of a host cell. They do this in a series of steps. First, the virus binds to a specific surface protein on the T cell called a CD4 molecule, as well as one of two co-receptors. The free virus is then able to fuse with the plasma membrane of the cell and empty its contents into the cell. At this point, the host cell has been infected. Reverse transcriptase from the host cell converts single strands of the viral RNA into double-stranded DNA. The newly formed strands of viral DNA travel to the host cell nucleus, where they combine with the host cell's own DNA through an enzyme called integrase. When the host cell is activated and divides, the viral DNA is read along with the host cell DNA, and long chains of proteins are assembled together to make new products. This assembly of viral particles forms an immature virus. The virus forces its way out of the cell, and in so doing, it takes some of the host cell membrane with it. Once free, the immature virus is free to go and infect another cell. Maturation of the virus occurs as an enzyme known as protease cuts the protein chain into individual proteins that form a functional, working virus.
Our product, Echinacea, is an herb that is used as an immune stimulant, antibacterial, and antiviral. It is taken to help with arthritis, genital herpes, immune support, otitis media, and upper respiratory infections. The most popular use of Echinacea is to help prevent or shorten the duration of the common cold. It is available over the counter in a variety of forms like capsules, tea, juice, and tinctures. It has been noted to cause immunosuppression after prolonged continuous use. It should not be taken with other immunosuppressants or corticosteroids, and caution should be used when it is taken with other medications metabolized by CYP3A4.
Scientists are unsure of the identity of all the active ingredients in Echinacea, and a clear mechanism of action has yet to be established. Heteroxylin and arabinogalactan are the two known active ingredients that play a role in its action. Echinacea acts by stimulating phagocytosis, increasing the release of cytokines and inhibiting hyaluronidase. Heteroxylaxin activates phagocytes, while arabinogalactan promotes the generation of T lymphocytes and increases the release of tumor necrosis factor, interleukin-1, and interferon beta-2 from macrophages. Overall, these actions may help to increase the body's resistance to infection. It's important to note that Echinacea does not appear to be either bacteriostatic or bactericidal. Echinacea can be administered orally, parenterally, or topically, but other specific pharmacokinetic data is not yet known.
Our group would not recommend Echinacea solely for the treatment of the herpes virus. The Natural Medicines Comprehensive Database rates this product as possibly ineffective for the treatment of the herpes virus. Currently, there is not a cure for herpes. However, there are several proven and FDA-approved products that can reduce the frequency, duration, and severity of outbreaks. These products include acyclovir and valcyclovir. Both of these are widely used today for the treatment of the herpes virus. So, in conclusion, our group would not recommend the use of Echinacea. Rather, we would recommend acyclovir, valcyclovir, or other FDA-approved products for the treatment of the herpes virus.